All right, you should be able to see my screen now. Which there's not much I'm showing you there, but let's go to here. All right, are there any questions before we begin? So today is October 3rd, and we're supposed to be covering chapter 1, finishing chapter 1, and beginning chapter 2. I hope we will finish chapter 1. We might have a bit to do. Please note that on Wednesday, there's going to be the first practice quiz. You can get extra credit for taking this quiz. I really want you to take the quiz for two reasons. First, you'll get familiar with my questions and how I ask questions and how the Canvas quizzes work. And second, you need to take Respondus Lockdown Browser. You need to use Respondus Lockdown Browser for all of the quizzes, except for Final A. But for all the quizzes in this course, you'll need to take the quiz with Respondus Lockdown Browser. So you need to download that and get it running on your system. And every once in a while, usually just one student every other term, there is a student who doesn't get the lockdown browser on their system. And if that happens, you need to contact the computer help desk. You can let me know that you're having trouble. And you should do this early, because if you wait until you take the quiz and then you contact the computer help desk and they're already shut because you started the quiz Wednesday night. Wait a minute. Yeah, Wednesday for the practice quiz. They may not get back to you until Thursday morning. And I will allow you to take a quiz late for about 2 days late. But after that, the answers will show. And then I will no longer allow you to take the quiz. I don't care what your excuse is. The answers are showing. You are not allowed to take an online quiz once the answers are showing. So you need to get Respondus Lockdown Browser up and running on your system to make sure that it works, because otherwise you're going to have trouble. And generally, about one student every other term has trouble with Respondus Lockdown Browser on their computer. They have to just change a setting on their computer. And the computer help desk is very good at getting the program working on your computer. I would not be able to help you change your computer settings to get Respondus Lockdown Browser working. So you need to contact them if you don't get it working. And you must get it working in time to take the quiz. Now, practice quiz is only an extra credit point, so that's not a big deal if you miss it. However, quiz 1 is going to be a regular quiz for 75 points. So you must get it running, the lockdown browser, on your computer before you take quiz 1. So take practice quiz 00, and then you'll know that the lockdown browser is working. Frequently, all the students who ever have any problem don't bother taking the practice quiz, and then they have trouble taking quiz 1. Any question about any of that? All right. Practice quiz 00 covers chapter 1 and chapter 10. It does cover the syllabus. It may cover lab 00. Let me look and take a look at an older syllabus, because I know I've got that written on there someplace, what that covers. And for some reason, I don't have it on this syllabus, and I just noticed that now. So let me see. The summer would have been the last term. Oops, that's the wrong one. So let me see if I added it there. No, I don't have it here. The last one would be spring 2003. There it is. Chapter 1, Lab 0, and Lab 1, and the syllabus. So let me copy that and put that on our syllabus. And I'll try and get that updated. Oops, that's not working right. Let's make that smaller. All right, any questions about the practice quiz? We are having a lab today. I'm going to discuss the infectious disease project. All right, if there's no questions, let's begin with chapter 1. Can somebody confirm? Is this the last slide we talked about last time? Did I go further in the lesson? Let me look and see if there's another 2 here marker. Nobody's saying anything, so I'm guessing you guys don't know either. This is the only
No,
map.
I think it was that one. Because you're talking about how microscopes got better through time yeah, it was this one.
Okay, great. All right. So the history of microbiology largely concerned whether or not life spontaneously generated or whether life came from another living organism. So that's the hypothesis of spontaneous generation, that life could spontaneously generate. And the other hypothesis is biogenesis, that life arises from a pre-existing life form. And from the 1600s to the 1800s, there was much debate in biology about these two hypotheses, especially in microbiology. The spontaneous generation believers believe that a vital force came into inanimate matter and then converted it into living matter. And of course, biogenesis, we all know a baby comes from 2 parents, and that one was never questioned. But microorganisms, they thought, could arise spontaneously. And over a century ago, there were beliefs and evidence for spontaneous generation. If you threw a log into the fire, sometimes a newt would crawl out of the fire, and people would say, spontaneous generation of the newt. Wood in the fire spontaneously generated the newt. Now, in reality, the newt just happened to be in the bark of the log, and it didn't like the temperature of the fire, so it crawled out. But people noticed that flies would emerge from manure, and once again they said spontaneous generation of the flies from the manure. And then maggots arising from decaying corpse. Once again, that was pointed to as evidence of spontaneous generation. Most were logical, but they were not scientific, and some were even amusing. Somebody wrote up a description where if you put sweaty underwear in a jar and put grains of wheat in the jar, it would spontaneously give rise to mice. In reality, the mice climbed into the jar to get the wheat, and then they were unable to climb out of the jar because it was glass. The mouse couldn't climb out of the jar. The mice were not spontaneously generated, and it had nothing to do with the sweaty underwear. I'm going to skip that slide. With microbes, people would get a tube of media and it would look clear like this with no microbes in it. And then with time, microbes would grow into it. And people said spontaneous generation of the microbes in the media. The vital force came into the media, converted it into living forms in the media. And this turbidity is evidence of growth, not of spontaneous generation. The first test of spontaneous generation was started by Francesco Redi, who you should know because not only did he give the first evidence against spontaneous generation, but he also was the first person to document a controlled scientific experiment where he set up an experiment, controlled the variables, and then wrote about it. So Francesco Redi was the first person to do that. And what he did was he got 3 jars and they were open and he put meat in them. And then he got 3 jars, he put meat in them and he completely sealed it. He noticed maggots appearing in the 3 open jars, the meat, maggot in the meat. But in the meat in the completely sealed jars, there were no maggots. Were those who believed in spontaneous generation? They said, well, you sealed the jar. You didn't allow the vital force to come in. So it doesn't prove anything. Well, then Francesco Redi took another jar, put meat in it, and covered the jar with a fine wire net. It was a metal net. And that kept the flies out. And there were no maggots in the meat, in the jars, covered with the wire net. So this gave the first evidence that first scientific evidence that spontaneous generation did not occur. And after Francesco Redi's time, you don't need to know the date, but after his experiment, people did not believe that large organisms like a maggot could spontaneously generate. However, they still believed that small microbes could spontaneously generate. Any question about any of that? About this same time were the first observations of microbes or the first study of microbiology. And that happened by Antony van Leeuwenhoek. I'm not Dutch, so I could be butchering his name. But he was actually an amateur biologist, microbiologist, and he was the best lens grinder of his day and the best microscope builder of his day. And he put all sorts of things under his microscope and then described microorganisms and teeth and scrapings from his teeth in rainwater, peppercorn infusions, and then samples of the River Thames, the river, the water source for London. And he saw microbes wherever he looked. This is actually a picture of his microscope. You'll notice it's a simple microscope. And like I said, he ground the lens himself. And he was such a good lens grinder, he was able to see things that other people could not see. It also helped that because he only had one lens, he could finely focus on a specimen. And with the compound microscopes of his days, they were blurry. So it would be very hard to see a microbe because they were so small and things were fairly blurry under the compound microscope. But he simply had a simple microscope, and all it is a super duper magnifying lens. He saw cells so small that they were not seen for another century. And here are his drawings, and you can recognize that's a bacilli. That's another bacilli. That's a spirillum. And these are rods. That's a bent rod. Oh, and the E is a cockeye. Sorry, I missed that. So he actually saw bacteria cells so small that they were not seen again for another 100 years. And it's just because his microscopes were so good. It took microscope builders 100 years before they ever improved the microscope that they would be good enough to focus in on bacteria. Any question about any of that? Around this time, Rudolf Virchow, you don't really need to know this too much, but he wrote up the theory of biogenesis. It was in belief long before he wrote it up, but he formalized it and published it. And you don't need to know the year that it was, but I guess it was 1858 is when he did it. But the arguments about spontaneous generation continued for several years after he published it. And the arguments continued until 1861. Once again, you don't need to know this year. But you should know that the arguments came to an end when Louis Pasteur set up an experiment and finally disproved spontaneous generation for microbes. He disproved spontaneous generation. First, Louis Pasteur showed that there are microbes in the air. And so this tube, where do I have it here? This tube, if it was open to the air or even exposed to the air, microbes could fall in and then begin growing in the tube. And he also would tell you that microbes were in the tube, although people could sterilize the tube. And if they left it open, microbes would get in it from the air. Now, let me go back. He also demonstrated that microbes could be destroyed by heat. So you could use heat to kill microbes. And then he did his ingenious, famous swan neck flask experiment to disprove spontaneous generation. What he did was he poured media in a flask that had a long glass neck, and then he heated the neck right here, and then bent the glass in the shape of a swan's neck, like that. And then he heated the media, sterilizing it, and then allowed it to cool. And he discovered that there were no microbes growing in the media, even though it was open to the outside, because the vital force could come in from this way and come in over the glass. The only time he got microbes growing in the media is if he poured the media down to here and then poured it back into the tube. What happened is the microbes would fall down from the air, and then they would stop here because of gravity. They would not go up the tube. And we actually have a flask like this outside the microbiology lab. If ever you're in the STEM building on the 3rd floor, there's a microbiology lab. In the window, there's a flask like this that was set up over 10 years ago. It might be over 15 years now. And there are no microbes growing in it, even though it's been sitting for 15 years. And eventually, all the media is going to dry out in the flask. But as far as I know, that flask is still there in the window. Any question about any of that? So that disproved that microbes could spontaneously generate, that they had to come from a pre-existing life form, meaning another microbe. Beginning with Pasteur's work, there were a number of discoveries, including the relationship between microbes and disease. That gave rise to the germ theory of disease, that germs could cause disease. This also gave rise to immunity, the fact that the body has an immune system and then can respond to germs making a patient immune to those germs. And it also gave rise to the antimicrobial drugs. We'll talk briefly about somebody who had the idea that we could find a chemical that could be toxic to the microbe and not toxic to us. And that led to the antimicrobial drugs. Any question about any of this? All right. After Pasteur's work, Pasteur went on and developed the basic concept of aseptic technique. You're learning that in the lab, even though you're not really practicing it. But if you keep a culture sterile by using aseptic technique, you can maintain only one organism in that culture. And Pasteur actually came up with the concept of developing a pure culture. It was a very difficult way. Sorry, I've got a fire engine. I don't know if you can hear that in the background. What he did was he did a bunch of dilutions and then replicated, I took a sample of that and put it into 10 tubes. And if only one of the tubes had growth and 9 of the tubes had no growth, he reasoned that one or very few cells caused the growth in that tube. And so the growth in that tube was likely to be pure, meaning from one or a few cells. And he was correct. And that was a way that him and early microbiologists came up with working with pure cultures. Koch developed that further, and he came up with a very easy way. Like I said, Louis Pasteur's way of getting a pure culture was very difficult. But the easy way was he simply streaked cells, bacteria cells, on a solid media. And what he did was he streaked for colony isolation. And if he took from an isolated colony and streaked it a second time on a plate, he then passed the cells through 2 colony formation events. And he reasoned that those cells that went through 2 colony formation events will likely be a pure culture. And he is correct that over 95% of the colonies that have been streaked out twice, meaning you streak it through 2 colony formation events, 95% of those colonies will be a pure culture. And that is called streaking for colony isolation. And like I said, Dr. Koch came up with that. In Dr. Koch's lab, one of his assistants developed nutrient agar, which was the first general purpose media, a media for growing many bacteria. It was a new media for growing microbial growth, and it was a very good media that could grow many microbes. It wouldn't grow something that's fastidious, but most other organisms that we grow in the lab, it would grow in nutrient agar. And then about this same time, Sergei Winogradsky developed the concept that microorganisms were involved in the biogeochemical cycles, such as the nitrogen cycle, what we've talked about, as well as the other cycles, like the sulfur cycle. The reason why Winogradsky was able to find this, discover this, and other people were not, was people were generally working with a pure culture. And if you were working with a pure culture, you could not have developed the nitrogen cycle because you need many different organisms living together to have the biogeochemical cycles like the nitrogen cycle. And Sergei Winogradsky studied communities of microbes. He didn't study pure cultures. Any question about any of that? All right, in the 19th century, you don't need to know exactly when, but Pasteur showed that microbes were responsible for fermentation. He studied how people made beer and wine, and found that it was because the microbe, the yeast, converted sugar to alcohol to make the beer and the wine, and that is alcoholic fermentation. He also then showed that other microbes could get in the beer or the wine, and they were responsible for spoiling the beer and the wine, as well as spoiling other foods. But he actually studied it first in beer. People were getting sour beer, and they asked Louis Pasteur to find out why. And he found that there were other microbes that were in the beer, and they would grow and convert the wine into vinegar, making the beer sour. So they convert the alcohol to acetic acid, which is the vinegar. Any question about any of that? Well, further on that work, Louis Pasteur developed pasteurization. He came up with a method to keep the beer from spoiling. So once it was fermented and was alcoholic beer, he then pasteurized it at a temperature that was high enough heat to kill the spoilage bacteria, but not high enough heat to evaporate all of the alcohol in the beer or the wine. And that we call pasteurization. It actually is very common in beer. Most beer is pasteurized. I only know of one exception, and that's Coors beer. They filter their beer. They don't pasteurize it. And it does taste different from what I hear. I'm not a beer connoisseur, so I don't know that myself, but my brother told me it tastes different. And pasteurization is very common in some of our foods. Most of you know it as milk. As far as I know, in most states, you cannot buy raw milk. You have to buy pasteurized milk. And it's heated to a temperature high enough to prevent the organisms from quickly spoiling the food, and then it kills the human pathogens that might be in the milk as well. And pasteurization is used in a wide variety of foods. Any question about any of that? So the concept of fermentation, that microbes can affect the organic matter and then spoil the food, such as beer, it led to the question, could microbes have a similar relationship with plants and animals? Might microorganisms cause disease in people as well as animals and plants? And this idea became known as the germ theory of disease. This was an important theory because originally people believed that breathing in poisonous vapors was responsible for at least some infectious diseases. Like it was believed that people who would go walking through a swamp would breathe in the poisonous vapors of the swamp and then come down with cholera. In reality, cholera was in the swamp, and then the people walking through the swamp picked up the cholera. And the cholera would be, I used to know this, Vibrio cholera, a bacteria which is bent rod, Vibrio. Any question about any of that? So the germ theory of disease came about, and there were various people like Ignaz Semmelweis, I could be butchering that name, and Joseph Lister, who came up with the concept that people should be sterilizing or cleaning their hands before doing things like surgery. And then you reduce the infection in patients. The germ theory of disease was not proposed by Dr. Koch, Robert Koch, but he did prove it. And what he did was he proved that the bacterium Bacillus anthracis causes the disease anthrax. He studied Bacillus anthracis, followed it in mice and found that it caused anthrax in the mice. And he provided the experimental steps he used to show that Bacillus anthracis causes the disease. And he then later wrote that up. And we call those Koch's postulates, the steps that you use to show that a given microbe causes a specific disease. Any question about any of that? So Koch postulate first reported that he showed that a specific bacteria caused disease. That was the first time that was ever shown. And he then wrote the steps that he used to show that, and that is known as Koch's postulates. This is the first time in this class we'll talk about Koch's postulates. We will talk about Koch's postulates 3 times in this class. That's how important it is. So you can expect questions on the quizzes about Koch's postulate as well as on the final. And by the way, I am probably butchering Dr. Koch's name. I'm pronouncing it as an English-speaking person. The Germans have a sound for CH, which is absent in the English language. And I can't really pronounce his correct name because I can't say how you do that. But it's a guttural X, okay? So I'm not even going to try to do it because I can't do it. Most English people call him Dr. Koch, but you could call him Dr. Koch, but most people say Dr. Koch. And the Germans say it correctly, but I'm not going to attempt to say it because I can't make that sound. Any question about any of that? All right. A little before Louis Pasteur's work, we had our first vaccine. It was created by Edward Jenner. You don't need to know when he did it in 1796, but you should know that the first vaccine was made by Edward Jenner. And what he did was he vaccinated against the disease smallpox using cowpox. Cowpox is another disease. It was a very mild disease in people, and generally only people who were handling the cows, like milkmaids, came down with cowpox. So it was a disease of cows. I don't know how serious the cowpox is among cows. I'm certain it wasn't as serious as smallpox was among people. And then he gave people the cowpox virus, and that protected them against smallpox. No one at the time understood why Edward Jenner's vaccine worked. And so there were no further vaccines for almost a century. It wasn't until the work by Louis Pasteur who showed that cowpox is a related virus to smallpox. And then when the body mounts an immune response to cowpox, some of it works against smallpox, and that provided the immunity to smallpox. With Louis Pasteur's work, there were other vaccines that were developed. Louis Pasteur made several vaccines himself. One of them that he's most famous for is he made the vaccine against rabies. Vaccination is actually a term derived from the word vacca, which is a word for cow. The protection that people get from a vaccine is called immunity. Any question about any of that? All right. Chemotherapy is the treatment of disease with chemicals. A chemotherapeutic agent is an agent you use against an infectious disease. Now, chemotherapy today is reserved for use against cancer. So this is a chemotherapeutic drug. Most people mean that to be a drug that we use against cancer. However, chemotherapy strictly means treatment of a disease, any disease with chemicals. Today, we don't use chemotherapy to use that word against microbial infections. We use the term antimicrobial drug or an antibiotic. Antibiotics can be synthetic drugs, meaning one that's man-made, or one that is made by a living organism. And this is a textbook definition, where an antibiotic is a chemical that is produced by a living organism, either a bacteria or a fungi, that inhibits or kills other microbes. This is not the way that antibiotics is used commonly in common usage by people. And by this definition, penicillin G would be an antibiotic. However, ampicillin, as well as carboxycillin, would not be an antibiotic because it is not made by an organism. It is man-made, or at least what's the word? Man changes it. Semi-synthetic is the word. So they would call it a synthetic drug or a, what do you call that? An antibacterial drug. That would be the textbook's definition for something like ampicillin. I won't use it that way because antibiotics in common usage means any drug which works against a bacterial infection. Any question about any of that? And most clinicians do use the term antibiotics as it's used in common usage, meaning they won't call penicillin G an antibiotic and ampicillin an antibacterial drug. The first drug that was ever available to treat a disease, which was actually quinine that people got from the bark of the cinchona tree. And this drug was used to treat malaria. Does anyone want to guess the year or approximate year that we had the first drug available to treat a disease, which was malaria? Come on, somebody guess. Even if you're wrong, that's fine. Nobody's going to guess. I'm just going to move on. All right. Paul Ehrlich came up with the concept that we could find a chemical to treat various human diseases. And what he did was he just took a chemical, tested it, didn't treat the disease, took another chemical, tested it, and on a sign outside of his lab, his workers put the sign, "Abandon all hope, ye who enter here," because they had to go through 500 chemicals before they found one that was successful in treating the disease syphilis. And he found this in 1910. He called it a magic bullet, a synthetic drug, an arsenic drug, salvarsan, which could treat a disease. And the disease he treated was syphilis. Any question about that? So salvarsan was the second drug we ever had available to treat a disease. Salvarsan isn't used anymore because it's fairly toxic. It's a synthetic arsenic drug. And we have antibiotics, which are not toxic to humans, or they might have side effects, mild side effects, but generally they're not toxic.
Can you either spell or type that drug that you're saying?
You're saying salvarsan?
Yeah,
Can you see the screen?
Salvarsan.
Yeah, can you see the screen? It's right here.
Oh, I see it right there. Yep,
Yeah,
thank you.
okay. I doubt I'm going to ask you a question. What was the name of the drug that Paul Ehrlich found that could treat syphilis? I would probably ask you a question more like, who was the person who discovered the first chemical that we found to be an antibacterial drug? And I'll use that wording because the first chemical was actually quinine from the bark of the cinchona tree. And we don't know who first discovered that, but malaria is not a bacterial drug. Okay. And I'll tell you the story of how that was found. It was actually found by Jesuit missionaries who went to Peru, and they noticed the natives eating the bark of the cinchona tree to treat the malaria. And they noticed that the patients were cured. And so they said this bark of the cinchona tree is really wonderful. It can treat malaria. And they took the bark of the cinchona tree back to Europe. And nobody wanted to guess when that was. And I'll tell you, it's much before 1910. You guys can look that up if you're curious. I'm not going to say if nobody guesses. All right. So Ehrlich came up with the concept of the birth of modern chemistry, where we could find a chemical that could cure a bacterial disease. And he came up with one to cure syphilis. You don't need to know the year. The next person to develop this, although I imagine Ehrlich came up with a few others after 1910 that were similar to salvarsan, but Alexander Fleming discovered the first antibiotic, a drug which was non-toxic to people, but toxic to bacteria. And he observed that what he did was he streaked out a plate, and he was a sloppy microbiologist, and he got contamination on his plate. And then he just left his plate on the counter rather than putting it in the incubator. And then he took off for the weekend. And then he came back on Monday and found the plate that he streaked out on the counter. And he looked at it and he saw growth on it and a colony of mold. And rather than throwing it away because it was contaminated, he looked at it carefully and he noticed that coming out of the colony of mold, Penicillium mold, there was a region around the mold where the bacteria would not grow. And he correctly concluded that something was made by the penicillium, which diffused through the agar and inhibited or killed the growth of the bacteria. And what that was penicillin G, the first antibiotic ever discovered. Unfortunately, Alexander Fleming was not able to purify penicillin G, and so it never became available to test on patients until the 1940s, where it was mass produced and purified and then given to the first two patients. If there's time, I'll tell you sometime about the story of the first two patients treated with the antibiotic penicillin G. So penicillin G was actually the second antibiotic that became clinically available to use in the hospital to treat a patient's bacterial infection. The first antibiotic that became clinically available was sulfonamide, a sulfa drug. And the reason is that this sulfa drug was man-made, and so people could make it and then purify it and then give it to their patients. Whereas the penicillin was grown by a a mold, and it took a fair amount of effort to grow it up and then purify it. And so it was not available until the 1940s, and 5 years or more earlier, sulfonamide became available in the clinics. Any question about any of that? All right, bacteria were once classified as plants, hence the term flora for microbes. If you ever hear me say the word microflora or flora, you can correct me. The trouble was I learned the term in microbiology before the modern term, and the microflora has now been replaced with microbiota. The normal microbiota are the microbes that are normally present in and on a patient or a human body. We'd call that the human normal microbiota. Microbes present in and on the human. But like I said, the term microbiota was not around when I took microbiota biology. The term was microflora. And it's an older term, it's not used, and hopefully I won't ever use it, but if I do, you can correct me. Animals and plants are host to a very large symbiotic microbial community where there are normal microbiota on all animals and plants. And these microbiota are central to the welfare of that animal and plant. The human normal microbiota is found usually only on and in the skin, the oral nasal cavities, the respiratory tract, and we're talking about the upper respiratory tract, the digestive tract, and the urethra. The normal microbiota is not normally found internal to the body, in the blood, the internal organs and tissues. The normal microbiota helps humans, prevents disease by growing in an area that a human pathogen will try to grow in. So they're competing for space and resources. Like if a normal a human pathogen were to fall on my skin, I've got lots of normal microbiota on my skin. So then the pathogen may not get established because it may fall on top of a bacteria, Staphylococcus epidermidis, and the human pathogen then may not grow. No space for it, and then no resources. The normal microbiota can compete for resources and prevent the human pathogen from growing. The normal microbiota can also secrete toxins that inhibit the growth of pathogens. We'll talk a little bit about this later. That's similar to an antibiotic, but it's a protein made by our normal microbiota, and it's very specific in what it can prevent. Usually it is very narrow, and it only prevents a few species or genera of bacteria from growing. An example of that would be E. coli can make this toxin that would prevent the growth of Salmonella and Shigella, but it would not prevent the growth of most other bacteria in your gut, such as, let me see if I can remember the name. No. Streptococcus? No. Enterococcus faecalis. That's a bacteria that all of us have in our intestines. And the toxin that's made by E. coli would not prevent the growth of that similar gram-negative bacteria, but it would prevent the growth of Salmonella and Shigella. And these are two human toxins, two human pathogens, sorry. The normal microbiota can also produce nutrients and growth factors for us. For example, a significant amount of our folic acid and vitamin K is made by our normal microbiota in our gut, making these items for us. And then the normal microbiota helps us digest our food. I think everybody knows that E. coli is required in our guts so that we can help digest our food. This is especially true for ruminants that eat cellulase, because no animal, no eukaryote for that matter, can digest cellulase. They have microbes in their gut which can digest the cellulase, digesting it into sugar, and then the microbe can make that sugar available to the cow or other ruminant. And that's why a cow can eat grass. They don't digest the grass themselves. They have it in their stomach, and they have 4 stomachs, and they have a microbe in their stomach that digests the grass. Don't try and eat grass because we don't have that microbe. We also don't have 4 stomachs. Any question about any of that? So the normal microbiota do not normally cause disease. In a healthy individual, the normal microbiota do not cause disease. However, they can cause disease in certain circumstances, such as when the patient is immunocompromised or their barriers have been disrupted. I'm talking about like a trauma wound where the skin is broken. So an infectious disease results when a pathogen overcomes the host resistance. And infectious diseases are caused by a pathogenic microbe. I already mentioned that a pathogen is a human pathogen is a microbe that causes human disease. Any question about any of that? All right, if not, let's briefly talk about emerging infectious diseases. These are new or changing diseases that are increasing or have the potential to increase in incidence. Does anybody know an emerging infectious disease? Come on. Nobody's going to suggest anything?
Would COVID be one?
Yes, COVID-19 would be an emerging infectious disease. And that's because before, I don't know, 3 or 4 years ago, I don't remember how long anymore, before 2019, humans had never seen COVID-19. So that is a new and emerging disease. Another one that they're watching is bird flu. It's a disease which causes the flu among birds, but it tends to infect people who are hanging around birds. I mean, you have to have close contact with birds, and then you can become infected. And it's very lethal. I don't know what the lethality rate is, but it's pretty high, 20%, maybe even as high as 50% of the people who come down with bird flu die. And their worry is that bird flu could evolve to spread among people. So far, that has not happened, that all the people who have gotten bird flu have gotten it from birds. And fortunately, it has never evolved to spread from person to person. But they're watching it because it could become a new disease that would increase in frequency. If ever it started changing and how it spreads person to person, that would be an emerging infectious disease. COVID-19 is just because it's a new disease. Before 2019, the number of COVID-19 cases in the entire world was 0. All right, that's the only thing you need to know about emerging infectious diseases, and that is the definition of what an emerging infectious disease is. I have more slides on emerging infectious diseases, but I'm not going to hold you responsible for these slides. They're just for your education and amusement. I will not quiz you over any of that matter, other than you should know what an emerging infectious disease is. Any question about chapter 1 and chapter 10? All right, let's move on to chapter 2. All right, this slide is showing you chapter 2, the chemical principles, listing the major goals and a rough outline. You should know the terms of this chapter. What's an organic compound and an inorganic compound? What is a hydrocarbon? What is a polar and a non-polar molecule? What is a hydrogen bond? What is an acid? What is a base? And what is the pH? You should also, with respect to the 4 major biological molecules, be able to describe the relationships and characteristics between the monomers and the polymers. To B, you should be able to describe the composition, structure, and function of the 4 major biological molecules. And you should be able to see, you should be able to give examples of each of the 4 major biological molecules. What are the 4 major biological molecules? You can find 3 of them on any box, like a box of cereal. I should say any box of food. What do you find on a box of cereal? Nobody's going to contribute. I'm not very pleased with that. You find carbohydrates, lipids, proteins, and those you find in all of the box of cereals. You also have nucleic acids. You won't find that on a box of cereal, but that is the 4th biological molecule. Any question about any of that? All right. Please understand the scale of things. I have this shown here. Let me blow this up. There's the size on the axis here, and then the listing of the organism. There is one mistake on the chart, and that's right here. They have 1 micrometer here and 1 micrometer there. This is 1 micrometer. That should be 1 millimeter. You should know the size of things. About the largest thing we'll discuss are people. Smaller than people are like a chicken egg. Smaller than a chicken egg is like a frog egg, and that's close to the smallest thing that humans can see with our naked eye. Smaller than that, we have eukaryote cells, and you need a light microscope to see them. And then smaller than a eukaryote is a bacterial cell. Most bacteria. You can get bacteria that are smaller than most bacteria. Generally about 4 micrometers is the smallest bacteria. This has it as 1 micrometer, 0.1 micrometer. So 0.4 micrometer is the smallest cell that we know is alive. The cells which are 0.1 micrometer probably are non-viable. And then smaller than a bacterium, we have a virus. And then smaller than a virus, we have large molecules like a protein. Oh, excuse me. We have I've got virus already, all right? Protein. And then smaller than a protein are the small molecules like water. And then smaller than a small molecule are the atoms. And you could go in reverse. The smallest thing we can see with a light microscope is an atom. You get atoms coming together to make small molecules and atoms to come together to make large molecules like a protein. Large molecules come together to make a virus. Large molecules come together to also make cells, but a virus is much smaller. And I'm talking about prokaryote cells. And then we'll learn at the very end that it might have been prokaryote cells that came together to form the first eukaryotic cell. And then eukaryote cells get together to make frog eggs, chicken eggs, and people. Any question about any of that? You do not need to know the size of something. Like most bacteria are 1 to about 5 micrometers long. You don't need to know that specific, but you should know that bacteria are smaller than eukaryotic cells, which are smaller than frog eggs, etc. Any question about any of that? So let's talk a little bit about the biological molecules. All of the biological molecules are an organic compound, or I could say an organic molecule. Organic molecules are typically complex structurally and how they're made. Organic molecules typically have carbon binding the molecule together. And that is partly because carbon can bind to 4 other molecules. Most of the bonds in a molecule are held together by covalent bonds, meaning a bond holding a substance together is a covalent bond. Occasionally, with 2 strands of DNA, you can also have hydrogen bonds. Organic compounds differ from inorganic compounds. In number 1, inorganic compounds typically lack carbon, but they do not always lack carbon. And organic compounds always have carbon. Inorganic compounds are required by living organisms for their growth, repair, and maintenance, as well as their reproduction. Some examples of an inorganic compound are oxygen in oxygen gas, and then hydrogen in hydrogen gas, as well as water and carbon dioxide. All of these are inorganic compounds. So carbon dioxide, oxygen, and water are all inorganic compounds. Water is one of the most important and abundant molecules in all living things. So let's talk a little bit more about water. Water is an inorganic compound. It is a small molecule consisting of only one oxygen and two hydrogens. Water is also a polar molecule where the hydrogens tend to have the electron less than the oxygen. So the electron that's just going around hydrogen and oxygen is going to spend more time around oxygen than it is around hydrogen. And that's just because oxygen pulls on the electrons much more intensely than hydrogen. Any question about any of that? All right. Within the water molecule, we only have a polar covalent bond. So this right here is a polar covalent bond. That's with the bonds within a water molecule. However, you can have bonds between different water molecules. And these bonds we call hydrogen bonds. So here is an oxygen, and it's going to have a partial negative charge because the electron will stay with the oxygen more than it will with these two hydrogen. So the molecule, or in this case, the element of oxygen, is going to be partially negative because that electron will spend more time with the oxygen than it will with the hydrogen. Conversely, the hydrogen will have the electron less time than the oxygen, so the hydrogen will be partially positively charged. And that's shown here, where the hydrogen is partially charged and the oxygen is partly negatively charged. Notice how I'm using the term partial charge or partly charged. Your textbook author doesn't use that term. And generally, he doesn't use the term as I'm using it, but he's a little more complex, and that's why I don't want to use our textbook author's terminology. They don't call it a partial charge. What's the author call it? It's not a complete charge. I don't remember what our author calls it. Any question about water? If not, let's watch a little video on the water molecule. Water gets together from oxygen and hydrogen, and there's the water molecule showing you the partial positive charge with the hydrogen and the partial negative charge with the oxygen. That allows a hydrogen bond to form between the partial positive hydrogen and the partial negative oxygen. That makes an electrostatic attraction, and that we call a hydrogen bond. It's a very weak bond. It breaks and forms all the time, which is why water is a liquid. And these bonds here break and reform all the time. so that this molecule can move around and this molecule can move around. And that's why, I don't know if you can see this, liquid water in here, why it moves. And that's because the different water molecules are moving over each other, sliding over each other. And the hydrogen bond is breaking and reforming all the time. And it doesn't have to reform here. This one can then form a hydrogen bond to another water molecule. And that's why we have liquid water. If ever you were to freeze the liquid water, these hydrogen bonds would not break and reform. They would be held in place, constant. And that we don't call liquid water. What do we call it?
Frozen?
Yeah, frozen water or ice, where these hydrogen bonds do not break. They're fixed and the water molecule is fixed in place. And of course, you can also break all of the hydrogen bonds by heating it, and then we don't have liquid water. We have the water molecules moving through the atmosphere, and that we call steam. Any question about any of that? And what happens is when the water molecule is at a high temperature, it will break that hydrogen bond and not reform it. And that's when we have steam. And then when the water molecule is very cold, has very little energy, the energy it has cannot break this hydrogen bond, and the water stays a solid piece of ice. Oh, here's what Tortora calls it. He doesn't call it a partial negative charge. He calls it a slight negative charge. And I just think the partial negative charge is a better term. And then a partial positive charge. It's not a complete charge because then it would be a +1 and a -1. And a -1 would have the electron totally and only at the oxygen, the extra electron. And then the +1 would lose that electron. In water, that doesn't happen. Okay, water is a good solvent. And that is because water can dissolve all polar molecules as well as ionic molecules. A polar molecule is a molecule like water that has a partial negative charge to the molecule and a partial positive charge to the molecule. The polarity of this molecule allows hydrogen bonds to form. And so like sugar is a polar molecule, and it'll have a partial positive charge on the sugar and a partial negative charge that can form hydrogen bonds. And in honey, we have that actually happening. Okay. In sugar, dry sugar, I don't think you have the hydrogen bonds forming. And they don't break and form all the time either. But in honey, they do. So that's what a polar molecule is. An ion is a molecule or atom that has a complete positive or negative charge. For example, sodium chloride crystal is shown here, and it is a crystal of a sodium ion, which has a +1, the green, and then a chloride ion, the bigger yellow molecule, which has a -1 electric charge. And how this has this charge is, let me see if I can remember now. The electron from sodium leaves sodium and then goes to chloride. So chloride picks up a -1 charge from the extra electron, and the sodium picks up a +1 charge because it loses an electron. You can have an ionic bond forming. We're not going to discuss that. And that's actually how the crystal of a sodium chloride crystal forms, and that is this positive charged sodium is attracted to the negative chloride ion, and that will form an ionic bond. Any question about any of that? All right. These ionic bonds can be broken by water because water has a partial charge, and it'll come up here. Let's talk about this one. Come up here, surround this chloride ion by putting the partially positive hydrogen against the completely -1 chloride ion. And then it'll surround this chloride ion and then bring it out into solution, where different water molecules will form around the chloride ion like this, where the partial hydrogens will be attracted to the -1 chloride ion. And that's an electrostatic attraction. The sodium likewise can have the same thing happening, where water will come around the sodium and then pull it into solution. The difference is that the partial negative oxygen will be attracted to the +1 sodium ion. And when this happens, we get salt water, where we mostly have water, but we have some salt dissolved in the water. Any question about water is a good solvent because it can dissolve all polar molecules as well as ionic molecules. A polar substance is one that will dissociate in water, forming a solute. And so, strictly speaking, we should say that this ionic crystal is not polar. But when we're talking it loosely, we will call anything that would dissolve in water a polar molecule. And by anything, we mean any polar molecule and any ionic molecule. Any question about any of that? All right. So let's finish this. A polar molecule are hydrophilic. That means they love being in water. Literally, they love water. That's what it translates to. But by hydrophilic, we simply mean that the molecule will dissolve in water. A non-polar molecule will be repelled by water, and that we say hydrophobic. Literally, they translate to fear of water. But what we mean is this molecule will not dissolve in water. For an example of a non-polar molecule would be oil mixed in vinegar. Vinegar is a polar substance that's mostly water, and you put the oil and the vinegar together, and they will separate because the oil is non-polar. It'll be repelled by the water. Any question about any of that? If there's no questions, I think it's time for me to end this. Let me check. Sorry, I just was teaching in the summer, and so my memory for, yeah, we're ending now. All right. Any questions? I'll see you later. Please remember, I do not use the chat box. So if that chat was for me, I don't even see it when I'm sharing my screen. All right. Bye. I'll see you in the lab.